Welcome to this presentation on the regulatory landscape in Canada for forced degradation studies. In this session, we will explore the role of ICH guidelines, Health Canada requirements, and how pharmaceutical laboratories ensure regulatory compliance. Forced degradation studies are a critical part of drug development, providing scientific evidence to support stability, safety, and quality throughout a product's life cycle. Forced degradation studies are intentional stress tests designed to evaluate how drug substances behave under extreme conditions. These studies go beyond routine stability testing by revealing potential degradation pathways and impurities. They play a vital role in ensuring patient safety, guiding formulation development, improving product design, and supporting regulatory submissions with strong scientific justification. Forced degradation studies typically involve exposing drug substances to various stress conditions. Heat accelerates degradation pathways, while light induces photolytic degradation. pH stress simulates acidic and basic environments, and humidity impacts moisture-sensitive compounds. Regulators emphasize that these studies are required not just to establish shelf life, but to demonstrate drug stability and safety. In Canada, forced degradation studies are governed by both international and national regulatory frameworks. Health Canada aligns closely with ICH guidelines while maintaining its own rigorous regulatory standards. Key guidelines include ICH Q1AR2 for stability testing and ICH Q3BR2 for impurity evaluation. Canadian laboratories must demonstrate strict adherence to these standards through comprehensive documentation and inspection readiness. The ICH requires a systematic and scientifically sound approach to forced degradation studies. These studies must evaluate all likely degradation pathways, identify and characterize degradation products, and generate data that informs formulation and stability strategies. Regulators assess whether studies demonstrate a true understanding of degradation mechanisms rather than relying on random or excessive stress testing. ICH guidelines specify that forced degradation studies should cover key degradation pathways. Photolytic studies assess light-induced degradation, while thermal studies evaluate stability under elevated temperatures. Hydrolytic studies examine degradation under acidic and basic pH conditions. And oxidative studies identify impurities formed due to oxidation. Together, these pathways provide a comprehensive degradation profile. Health Canada integrates forced degradation data into the overall regulatory evaluation of drug submissions. These studies are reviewed for scientific soundness, reproducibility, and complete documentation. Inadequate or unexplained degradation findings can lead to regulatory delays, additional study requests, or inspection observations. A systematic and well-documented approach supports a smoother approval process. Laboratories design forced degradation studies through a structured process. This begins with risk assessment to identify likely degradation pathways, followed by careful study design and controlled sample preparation. Labs must balance inducing sufficient degradation while maintaining sample integrity. Clearly defined acceptance criteria, appropriate controls, and reference standards are essential for regulatory credibility. After stress testing, analytical techniques such as high-performance liquid chromatography, LCMS, and UV visible spectroscopy are used to detect and identify degradation products. Regulatory agencies require validated methods capable of detecting all relevant impurities with accuracy and precision. Equally important is complete documentation, including SOPs, lab notebooks, and study protocols to ensure traceability and inspection readiness. In conclusion, forced degradation studies are not just regulatory requirements, but powerful scientific tools. They enhance understanding of molecular behavior, protect patient safety, and strengthen regulatory submissions. By following ICH guidelines, meeting Health Canada expectations, and maintaining robust documentation practices, laboratories build confidence in product quality. Compliance, combined with strong science, leads to successful regulatory outcomes.